And let's continue with this beautiful pathway with talk from Sodbo Sharapov from the same Institute of Cytology and Genetics, speaking about, again, about mapping genes involved in control of, continuing basically about Nglycosylation of blood glycoproteins. And Sodbo is joining us from Novosibirsk. This is OPSI probably, and some pine trees or whatever. Okay, Sodbo, the floor is yours, so welcome.
Yeah, thank you very much. So can you hear me well?
Fine, yeah, go ahead.
Yeah, great, thank you. Yeah, it's a tough time here, so global warming, etc. So the summer in Siberia becoming like this on my screen. Yeah, so in my talk, I will continue the line of the story of mapping genes controlling the Nglycosylation of proteins. And if Yuri talked about the genetic control of Nglycosylation of a single protein, immunoglobulin G, in my talk, I will tell you the story about the genetic regulation of Nglycosylation of all human blood plasma proteins. So to begin with a short reminder that the glycosylation is an enzymatic addition of a carbohydrate, so-called glycan, to a protein, and this glycan, as a part of a glycoprotein, affects the physical properties of glycoprotein and its biological functioning. And this post-translational modification is indeed quite abundant. So around 40% by mass of all human blood plasma proteins are Nglycosylated, and majority of these glycoproteins are secreted to a bloodstream by two tissues. It's a bone marrow that secretes the immunoglobulins and liver that secretes a variety of different Nglycoproteins. And the blood plasma and glycome is one of the most studied objects in human glycomics, partially because there are technologies for high throughput profiling of plasma and glycome. And one of the most commonly used one is the ultra high performance liquid chromatography, or UHPLC in short. So on the slide, you can see a typical representative chromatogram, which separates nicely different N-glycans, which, as you can see, are irregular polymers, branched irregular polymers consisted of several monomers. And below you can see the different kinds of glycoproteins to which these N-glycans are attached. Such technologies as UHPLC allowed researchers to conduct well-powered population-based studies of human plasma N-glycome variation. And as you learned from Yuri's talk, we know now that this variation is heritable. We know that this variation is associated with a risk of many of human diseases. However, we lack of interpretation of these associations. We, for example, even do not know the direction of this association. And in partially, this is caused by the fact that, as Yuri told that from biochemical point of view, the biosynthesis of N-glycans is well known. However, very little is known about the in vivo regulation of this process. So in our study, we aimed again to define novel regulators of Nglycosylation of blood plasma proteins by doing the GWAS. So we, with our collaborators, collected the largest collection of samples. It's around 11,000, almost 11,000 of samples from 7 cohorts. All these samples were profiled by the N-glycome of these samples were profiled by UHPLC, and of course these samples were genotyped by Mill and some SNPs. We tested association between each trait and SNP. We identified loci, and of course we confirmed these associations in independent samples. And finally, we did the gene prioritization, as GWAS do not directly pick up the causative gene. So what we have got? In our meta-analysis, we identified and confirmed association of 31 loci. 16 of these loci are novel, so they have now been found in the total plasma N-glycome GWAS. Overall, the found signals explained up to 21% of our traits variation, which can be translated into up to 36 explained percent of SNP-based heritability explained. So this is the loci. Of course, we did the candidate gene prioritization analysis. This is a tough task, as you may learn from the Shamil Tunayev talk and from Yuri's talk. We applied a variety of approaches. So in the end, finally, we prioritize the candidate genes in the found loci. So in the next slide, I will talk a bit more about these genes using this nice graph. So this is the glycop genome association network. Here you can see the squares and circles. Squares represents the loci which we have found and confirmed, and circles represents the N-glycan traits that showed the genome-wide significant associations with this loci. So we have 31 squares and approximately 100 N-glycan traits here. So first, as in previous talk, we see 8 loci that contains the glycosyltransferases genes, which codes these enzymes with known role in N-glycan biosynthesis. So we know these enzymes, we know their enzymatic activity, and as a proof of principle, we see that the biochemical structure of N-glycan traits associated with each of the glycosyltransferases, they are in line with enzymatic activity of these enzymes. So next, we see 2 loci that are coding the N-glycoproteins. We see locus IGH, which codes the heavy chain of immunoglobulin G. And we see the locus with haptoglobin, which codes, surprisingly, the amino acid sequence of haptoglobin protein, which is quite abundant in human blood plasma and glycome. So therefore, these loci might be associated with N-glycosylation either through controlling the level of the glycoprotein in blood plasma or through, for example, disturbing the N-glycosylation site on this protein. Of course, we see loci with transcriptional factors. So we see 9 loci marked by green. So there are different kinds of transcriptional factors, and some of them specifically works in immune cells. Some of them works, for example, as HNF in liver. And then as Yuri told in his talk, we see that some of the loci with transcriptional factors affect the same glycans as some of the glycosyltransferases loci. So we can speculate that these transcriptional factors may affect the expression level of these glycosyltransferases gene. And finally, we colored the N-glycan traits in several colors. So basically, how we classified these N-glycans? We classified them into 4 baskets. So the first basket is N-glycan traits that are linked to N-glycoproteins secreted by bone marrow. So it's mostly immunoglobulins. In the second basket, which we colored by blue, we put the N-glycans that are linked to N-glycoproteins mostly secreted by the liver. Purple color means these N-glycans are linked to both type of proteins. And we also have gray circles, which are, let's say, dark matter. For us, we cannot say explicitly the source of these N-glycans. And when you look at this network, you can see that it can be quite nicely divided into 2 subnetworks, the lower and the lower one and the upper one, which are partially overlapping. And what is interesting, so the upper network consists of N-glycan traits that are secreted by liver. And here we see this HNF1 alpha transcriptional factor, which liver-specific transcriptional factor. And in the lower network, we see N-glycan trace that are secreted to a bloodstream by bone marrow, by plasma cells. And here we see a lot of transcriptional factors that are specific to the immune cells. And moreover, almost all of the genes from the lower network were found in the genome-wide association study of glycosylation of immunoglobulin G. So we can conclude that there are two tissue-specific subnetworks of regulation of N-glycosylation. And what else we can say that if you want to, if somebody wants to study these loci and their effects on the N-glycosylation, that in vivo or in vitro, they should, here for these genes, they should use the hepatocytes derived cells, for example, HepG2 or whatever. And for this network, we need to use the cell lines which are close to immune cells. For example, I don't know, LCL or etc. So as a conclusion, we can say that we identified and confirmed cessation of 16 novel loci. In these loci, we suggested the candidate genes for further functional studies. And also using this nice network, we probably identified the tissue-specific gene networks that regulates the N-glycosylation. And again, thanks for our Russian Science Foundation who's funded this study and for all our team. And of course, this work couldn't be done without our collaborators. Thank you, and I'm open for your questions.
Thank you. Okay, nothing in the internet yet. Anybody from the audience? 1, 2. Okay. Yeah, Sinan, so probably that's it. Thank you, Sodbo.
Yeah, thank you.
Bye-bye. Thanks a lot.
Yeah, bye.